Hi viewers, today we are going to discuss the hypersensitivity reactions. Under this topic, we are going to discuss definition and what are the allergens, classification of the hypersensitivity reactions, general mechanisms beyond all the reactions, specifically type 1 hypersensitivity and the steps involved in the type 1 hypersensitivity and the related examples. Here, when an individual has been immunologically primed, that is exposed to the antigen, the further contact with the same antigen leads to secondary boosting of immune response, due to which a severe damage is caused to the host tissue. Therefore, this type of sudden harmful response is known as the hypersensitivity, or commonly called as the allergy. And the substances which are capable to cause allergy are commonly called as allergens. These allergens are basically antigens like dust particles, pollen grains, fungal spores, microbial eggs, drugs, altered food, certain edible oils, and the polluted water. These allergens show some common characteristic features, like these all are capable to cause asthma, eczema, itching, sneezing, sudden motions, vomiting, skin rashes, and the inflammation, otherwise the abscess, and the lesions in the various kinds of tissues. It should be remembered that hypersensitivity reactions differ from protective immune reactions only in that they are exaggerated or inappropriate and damaging the host tissue. The cellular and the molecular mechanisms of these two reactions are virtually identical. This should be remembered. And coming to the classification of hypersensitivity, Coombs and Gell, they classified the hypersensitivity reactions into four forms, namely type 1, type 2, type 3, and type 4. The classification is useful guide to understanding the different forms of the response given by the immune system. In general, the type 1 hypersensitivity reaction is known as the anaphylactic hypersensitivity. Type 2 is antibody dependent cytotoxic hypersensitivity. Type 3 is immune complex mediated hypersensitivity. And type 4 is known as cell mediated or delayed type of hypersensitivity. So these all are the classifications done by Coombs and Gell. Coombs and Gell were the scientists who classified the hypersensitivity reactions. Let us see the general mechanisms behind the hypersensitivity reactions. Start from the type 1. Here, anaphylactic reactions are mediated by IgE antibodies, which bind through the Fc portion to the receptors on mast cells and basophils. When cross-linked by antigens, the IgE antibodies trigger the mast cells and basophils to release pharmacologically or biologically active agents that are responsible for characteristic symptoms of anaphylaxis. The reactions are rapid, occurring within minutes after challenge, then that is exposure to the antigens, so that they are also known as the immediate anaphylactic reactions, immediate hypersensitivity. And coming to the type 2, these are the cytotoxic reactions occur when IgM or IgG antibodies bind to antigen on the surface of cells and activate the complement cascade, which culminates in destruction of the cells. Whereas type 3, the immune complex mediated reactions occur when complexes of antigen and IgM or IgG antibody accumulated in the circulation or in the tissue and activate the complement cascade. Granulocytes are attached to the site of activation and damage results from the release of lytic enzymes from their granules. Reactions occur within hours after the entry of the antigen. And then coming to the fourth one, type 4, cell-mediated immunity or delayed type of hypersensitivity is mediated by T cells rather than by antibody. On activation, the T cells release lymphokines that cause accumulation and activation of macrophages, which in turn cause local damage. This type of reaction has a delayed onset and occurs 1 to 2 days after the challenge with the antigen, so that it is known as delayed hypersensitivity. Let us see the first kind of hypersensitivity, that is type 1 anaphylactic hypersensitivity. Anaphylactic hypersensitivity. Type 1 or anaphylactic hypersensitivity reactions always mediate with the, see here, ana means away from. Ana means away from. Phylaxis is the protection. Whenever you get the anaphylactic hypersensitivity, that leads to the away from protection. There is no protection provided by the immune system. This was the Greek term coined by two scientists, namely Poriter and the Richet. Poriter and the Richet. Type 1 or the anaphylactic hypersensitivity is also called as the immediate hypersensitivity. It is characterized by the production of IgE antibodies against foreign proteins or foreign antigens that are commonly present in the environment. For example, the pollens, animal danders, or dust, otherwise the mites. These antibodies bind specifically to the high affinity receptors on the surface of the mast cells and the basophils, which are the only human cells that contain histamine. Subsequent exposure to the same antigen will lead to a rapid release of histamines, and other mediators include leukotrienes and the cytokines. These all are commonly called as the biologically active substances. The conditions that are associated with the type 1 hypersensitivity include asthma, hay fever, and atopic dermatitis. The symptoms vary and depend on the exposed tissue site, but most common features are reddening of the skin, which is called as the erythema, and soft swelling of tissue due to fluid accumulation, which is commonly called as the edema. So these are the general features of the anaphylactic reactions. See here we are observing, this is the mast cell and this is the basophil cell, and both of them, they have a specific receptor for the immunoglobulin E, Fc portion of the immunoglobulin E, to which actually immunoglobulin E binds and forms the anaphylactic reactions. So the mast cell and basophils, they are similar and they have the histamine. Whenever they blast due to the activation of the IgE, they release all these biologically active substances into the body fluids. Now we will discuss the different phases, otherwise the steps involved in the development of anaphylactic hypersensitivity. Now the type 1 hypersensitivity is having three phases, namely sensitization phase, activation phase, and the effector phase. Let us see one by one. Sensitization phase. What is the sensitization phase is IgE antibody is produced in response to an antigenic stimulus and binds to specific receptors on mast cells and basophils. All normal individuals can make IgE antibody specific for a variety of antigens. Generally, those are the airborne antigens, otherwise the allergens. A response is encountered only on mucosal surfaces such as lining of the nose, lungs, and the conjunctiva of the eye. IgE production is T cell dependent. Why? Because interleukin 4 is produced by CD4 T helper cells, which is important requirement for the IgE production. Suppressor T lymphocytes, interferon gamma decrease the IgE production, thus balance is maintained between those factors. The entry of allergens disturb the balance and stimulate IgE producing B cells. IgE antibody is made in small amounts and very rapidly becomes attached to mast cells and basophils as it circulates past them. Mast cells found abundantly around the blood vessels, in connective tissue, lining of gut, and the lungs. IgE has been produced in an individual is considered to be sensitized individual. So that is a sensitized phase. And coming to the activation phase, a re-exposure to the antigen triggers the mast cell and basophils to respond by release of the contents of their granules. The activation phase of type 1 hypersensitivity reaction begins with the triggering of mast cell to release its granules and their pharmacologically active contents. It requires that at least two of the receptors for the Fc portion of the IgE molecule. In the simplest and immunologically relevant manner, this linkage is accomplished by a multivalent antigen that can bind a different molecules of IgE to each of several epitopes on its surface, thus cross-linking them and effectively triggering the cell to respond. The triggering of a mast cell by the bridging of its receptors initiates a rapid and complex series of events culminating in the degranulation of the mast cell and the release of pharmacologically potent molecules. By series of events, the mast cell granules are moved by microfilaments to the cell surface where their membrane fuse with the cell membrane and all contents are released to the exterior. Here we will observe the first exposure of the allergen, and thereby here there is a processing and presentation of the antigen by the antigen presenting cell. This peptide absorbed by the T helper cell and it is T dependent activity, thereby B cell get activated and produce IgE kind of antibodies. Here the same antigen is re-entered the body, re-exposure, that leads to the secondary response where the IgE rapidly produced and contact with the at least two antibodies on the nearest adjacent Fc portions, and thereby there is a degranulation of the mast cells or basophils takes place. And coming to the effector phase, where a complex response, otherwise we should call it as the anaphylaxis, occurs as a result of the effects of the many pharmacological active agents released by mast cells and the basophils. So here these all are the pharmacological active substances which are released outside. Basically they are the pharmacologically or biologically active substances of the mast cell, otherwise the basophil cell. They include histamine, leukotrienes, heparin, prostaglandins, proteolytic enzymes, serotonin, and the thromboxane like substances. These mediators trigger and cause smooth muscle contractions, vasodilation, increased vascular permeability, and mucus secretions. These cause the finally they cause the vasodilation and wheal and flare, edema, redness, and as well as the itching. So these are the things to be caused by the biologically active substances. The anaphylactic reactions are categorized into two groups, namely systemic anaphylaxis and the localized anaphylaxis. Systemic anaphylaxis that deals with it is a generalized response that occurs when an individual already sensitized to an allergen receives a subsequent exposure to it. For this, the best example is the respiratory impairment caused by the constriction of smooth muscles in bronchioles. This condition in severity results in reduced blood pressure and asphyxiation and causes death of the individual. The most common allergens responsible for systemic anaphylaxis includes drugs like penicillin, passively administered antisera, and insect venom. Whereas if you observe the localized anaphylaxis, it is referred to as atopic allergy or atopy. The symptoms of atopy are primarily depend on the route of entry of allergens into the body. Hay fever or allergic rhinitis is a good example of an atopic allergy of upper respiratory tract. The bronchial asthma is an example of an atopic allergy involving the lower respiratory tract. The common allergens that produce hay fever as well as bronchial asthma are plant pollen, fungal spores, and the animal dander and house dust mites. These all are capable to cause the localized anaphylaxis. This is all about the type 1 hypersensitivity, which is usually called as the anaphylactic hypersensitivity reactions. Thank you one and all.